So hi everyone. I hope you can hear me properly. So today, as the name suggests, we are going to talk about the blotting techniques. Okay. So blotting techniques is the important concept that we need to know about know for IIT-JAM. So it falls under biotechnology, and the subtopic is biological techniques that are used for research. As a student who is going from a transition to a bachelor's to a master's, they need to learn this topic from their courses, and they really need it, right? So we're going to talk about this today. Okay, so hi everyone, whoever is online. Now let's talk about these technique, but before that I wanted to tell you something and at least give you some idea what are the important aspects and maybe clear some of your doubts. So most of you guys who haven't subscribed the channel yet, that means Unacademy's YouTube live channel, so hit that subscribe button and click on the bell icon for getting notification, and don't forget to share in all the social medias such that your friends can get benefit out of it. It's totally free, right? These lectures are aimed for those people who can't afford simply good education, and that is why I'm bringing up quite a lot of free contents that you guys can use for your educational purposes. Yep. So let's begin. Most of you guys ask these questions, and I get these questions very often. So before starting the class, let's talk about these questions a little bit and tell you about some good announcements, then we start the class. So guys, you guys ask that what are the best resources? Do I need coaching or self-study is sufficient? You also ask that on which part of the biology I should focus on, how to manage my time, and exactly like how to get a single digit rank. So all of these questions can be answered if you take my Unacademy Plus course. Now you would be saying, why should I even take my Unacademy Plus course, right? So the reason is the following: because you mostly go to your colleges. Sometimes all of the classes in the college is not good enough. So you might think that this is boring, and you wish that classes could be more engaging, more interesting, more interactive, but that doesn't happen. So in order to prepare better, you go to tuition centers, and what happens? They charge you a bomb. It really burns your pocket, and you end up getting a huge headache. Is there any solution for that? Of course there is. Introducing Unacademy, because here the courses are online. Especially during this lockdown period, you can access this course from anywhere you want. You just need to have an internet connection. You can even download these courses. So you could have free courses running in YouTube platform, or you could have free courses in Unacademy education app, and you have to pay a little bit amount for the plus subscriptions, which are more structured course, guys. And if you haven't yet subscribed to the YouTube channel, subscribe, because you are going to get free contents there. Okay, so there are two type of classes. There are free classes, there are paid classes. Obviously, if you have interest and you can't afford the paid classes, you can definitely join the free classes. But let me tell you a little bit about the paid classes. The paid classes, you're paying a very little amount every month. You're paying 2000 rupees per month, but yeah, that is reduced to 1800 when you use a coupon code of AP10. That would give you a 10% discount. But let's say you take a 12-month subscription, then the money is even more reduced, like it's like 46 rupees per class. Okay, so let me give you an update about what courses are running right now in Unacademy Plus platform. So these are the courses which are finished and the courses that are ongoing right now. Giving a code AP10 would give you 10% discount. Now let me tell you a little bit about the benefit of these courses, because all of these courses have very nice visual, good handwritten notes are provided, interactive flashcard would augment your learning process. So why are you waiting for? Register now. But you should always try before you buy. So that is why today we are going to learn about or get a glimpse about what type of questions are asked. So hi Ashutosh, hi Vivash. So today we're going to learn about the blotting technique. So without wasting further time, we might start about the blotting technique, right? So let's start. I hope some of you guys would join the plus course and we'll discuss more. So the question is, what are the important concepts? What are the questions that are asked from this blotting, this particular concept, right? So in order to know that, you have to watch this video till the end, because at the end I'm going to discuss in details that what are the important questions that you might face in IIT-JAM exam or any other MSc level entrance exam. So today we are going to at least learn 2-3 blotting process. First we are going to start with Western blot, right? Okay? So let's talk about Western blot. So Western blot, whenever we talk about any concepts or any biological techniques which are important for IIT-JAM, what we really need to do is to understand the principle. Second, we need to understand the usage of it. So all of these things are very important, right? So let's talk about that. So Western blot is a technique by which you can detect proteins, your protein of interest, and you can do a lot of things, like a lot of other applications are there for Western blot. So let's start to understand the bits and pieces of Western blot, and then we can understand what is the usage of it. So let's start. So the first step is to run a SDS page gel. So SDS page gel is very similar to agarose gel, only the material is different here. So proteins are normally run in a SDS gel. So what you do after running a SDS gel is to get the gel, and you stain the gel with Kumasi blue. So I have a different video on SDS page which would be uploaded soon, maybe sometimes. But here you see, after you do the SDS page gel, let us assume that you know SDS page gel, how SDS page work and all of these things. So then what you do is take out the gel, blot it, right? So you have a lot of bands in the gel. You blot all the bands in a membrane, right? A PVDF membrane. But the problem is, in case of gel, you might have a protein which is 75 kD, but you might have another protein which is also 75 kD. You can have a bunch of them. So only having one particular band at a desired size does not tell you that this is your protein of interest. This could be your protein of interest, but along with it, it could be some other non-specific proteins. So that is why Western blot is important, right? So that is why we need to understand about Western blot, how we can determine our protein of interest using Western blot. So again, Western blot is also dependent upon antigen-antibody interaction. So you might think it as an application of immune technique. So as I was saying, that this particular 75 kilodalton band, assuming like this is a protein of interest, so we have to blot our gel. And after blotting, what would happen? Like when you put the membrane on it and blot it with a electrical charge transfer, what happens is all of these proteins goes onto the PVDF membrane, right? Now on the PVDF membrane, you have to probe. Hey Shubhadeep, hey Sandeep, sorry, not Shubhadeep, Sandeep, hi. I see a couple of new people are joining. Okay, so now you have changed, I mean, you have like blotted the proteins from the gel to the PVDF membrane. Now, what is the benefit with that? Like, why you want to do that? What was wrong with the gel? Because simply the gel is not so easy to handle, and like, how would you do the further steps in the gel, because it's pretty flimsy. But the membrane is far more rigid and it would bind to the protein firmly. So that is why we use this particular PVDF membrane. Now what happens in Western blot is simply you have your protein of interest. So imagine this particular protein is the particular protein you want to detect. Again, I'm going to tell you about exact usage of Western blot in research, or how it is important for biology. So we'll talk about it, why this technique was developed. So ultimately, the antigen-antibody interaction is very important. So let's say you have an antibody of your choice which specifically detects the protein of interest and does not detect any other protein. So it might be a monoclonal antibody in this particular case. After that, you detect that particular monoclonal antibody interaction with your antigen. By what? by several techniques such as your colorimetric techniques, where substrate is giving rise to a colored product. You can also use chemiluminescence technique or fluorescence technique to detect whether the protein is present or not. So even if the primary antibody binds to the protein of choice, but we might not be able to see it because there is no detection method, right? So that is why you need a secondary antibody which is sort of linked with any kind of detection system. That is important. So this is the crux of these Western blot techniques. So this is how, using immune interactions, that means antigen antibody interaction, you can determine whether a protein of interest is present or not. So look at it. Here there would be lots and lots of protein, right? There could be some protein which are pretty similar in size, like for example this particular protein and this particular protein might be similar in size, but you are detecting only your protein of interest because you have given an antibody for as a probe to detect your protein, but not other proteins, right? So immediately it would tell this particular technique, the power of this technique is to determine whether your protein of interest is present in a particular biological sample or not. Now pause for a moment, think that whether you can use this technique for any other use or not. For example, biomedical diagnostics or biomedical research, right? So let's try to understand that and take some examples, right? For example, I was telling you Western blot could be hypothetically used to diagnose viral diseases like AIDS or even coronavirus diseases, but there are certain limitations to it because of the cost and exactly availability of the antibodies. But you can clearly understand, let's say you have a patient sample, from there you can extract the proteins, you can run it in the SDS page gel, followed by you can separate them in the gel, blot these gels, right, and ultimately look for the viral antigen using specific antibody. For example, in HIV, the particular antigen which is most popular and people look for this particular antigen is GP120. So definitely you can look for GP120 in your blood sample, and definitely using different techniques like different secondary detection techniques, you can develop your blot, and that would tell you whether you have AIDS or not, whether you are infected by a virus or not. So these kind of diagnostic purposes can be served using Western blot. So that is one of the particular use of Western blot. I hope you guys are understanding. Yep. So here Shushrita Acharya has asked something, that whether I'm providing a BT course or not. So I have a BT course in Unacademy, so it is already there, and also many of the free courses would be coming in the YouTube platform and also in their learning app. So yes, there are. Okay, so Western blot can be used for a variety of usage, right? So for example, Western blot can be used to study cell signaling. So in my other videos, I have already talked about cell signaling. So let's try to understand that how Western blot can be used to study cell signaling. Okay, so very interesting. So let's imagine a pathway in our head. So we all know about receptor tyrosine kinase and MAP kinase pathway. So we know in MAP kinase pathway, the key event is the kinase cascade, like one particular kinase phosphorylated substrate. So there is phosphorylation event. So imagine this is a particular kinase which is phosphorylating its substrate. This particular blue protein is phosphorylated by this violet protein, and as a result a phosphorylated blue protein is generated. So this is one of the readout of the signaling pathway. That means if we can determine that this phosphorylation event took place or not, then we would have an idea. That the signaling is on or not, and this can be determined by Western blot. Amazing, right? So we pretty much look at Western blot as a blotting technique. It's used to determine a protein of interest or not. But what I want you guys to think about is in what possible circumstances you can use Western blot, right? And how you can use Western blot in biomedical research or maybe in diagnostic labs and all of these things. So it's a widely used tool, right? So in this example, I said that Western blot can be used to study cell signaling, and I showed you that, I mean, let's imagine this pathway where a phosphorylation is a readout of the signaling, and what you do using Western blot. So you know, if you have an antibody which is specific for an unphosphorylated version of the protein, and you have another antibody which is dedicated to the phosphorylated version of the protein. So you need to have that antibody. That is the assumption that you need to have that antibody in your hand, or you have to generate it. So in my other videos, I would also tell you how antibodies are generated. I'll tell you a story. But anyway, so if you have that particular antibody in your hand that detects a phosphorylated protein and another antibody which detects unphosphorylated protein, then what you can do using Western blot, you can check the ratio. So in this particular case, what is happening in the blot, you first see this particular case. In this particular band is corresponding to the unphosphorylated protein, right? And this particular band should be the phosphorylated protein. Initially, you don't see any kind of phosphorylated protein, but later on you see a band and you see a reduction in the unphosphorylated population. That means here in this particular second lane, you can see a phosphorylation event is happening. I mean, definitely you are seeing some bands on a blot, but definitely this would tell you that this phosphorylation event has happened, and as a result the signaling is on, the signaling is turned on, right? So these are few important aspects or few important usage of Western blot. So wait a second, we pretty much learned what? We pretty much looked at how Western blot works, how SDS page and Western blot is pretty much coupled to each other, and what is the principle of Western blot, how it works on the basis of antigen antibody interactions. Then what we learned? We also learned that what would be the usage of Western blot. As a scientist, maybe how we can use Western blot to study various aspects of biology. Or we also looked at as a diagnostic tool, Western blot can also be used. I mean, forget about the cost, but hypothetically it can be also used, right? So these are the all possible application for Western blot. So for example, if you face a question in IIT-JAM that Western blot in one column and other column is try to detect your protein of interest, or to study a signaling pathway, or to diagnose some viral diseases. So all of these kind of questions can be asked, right, in the IIT-JAM question paper. For example, in one column they said, okay, these are the column of technique names, okay. This is the common question that are asked, and these are damn easy to answer. So most of the cases these are very scoring, so that's why I am telling you guys. So you just need to know the principle, and these illustrated videos would really help you to understand the principle of these techniques, and it would be really easy to understand with good visualization, right guys? Okay, so pretty much we get a summary that we have to start with our SDS page gel. Then we have to finally take out our gel, we have to blot it, and then ultimately we are going to look for whatever we want. Anyway guys, we are pretty much done with the Western blot part. Let's talk about another blotting technique. So this blotting technique is known as northern blot. Now northern blot could be used to detect RNA of interest in your sample. Now these days, let me tell you frankly, northern blot is not that much used, but since it was a very important technique in 1970s, so let us talk about these things, and we'll also talk about what is the alternative for northern blot and what people are using these days. We would also look at the application of the northern blot, and ultimately we are going to discuss some questions on northern blot. So let's talk about it. So let's say you have a cell, okay. So from that cell you have to isolate RNA, and you want to understand that your RNA of interest is present in this particular sample or not. So let's say you have treated the cell with some drugs and your expectation is a gene A would be expressed, so gene A mRNA would be detected. So you are going to probe for gene A mRNA, right? So you definitely need probe. So what is common for all of these blotting technique, that this is a detection system, like either you use some antigen antibody interaction to detect protein of choice or protein of interest, and here you are using the probe based approach, like you are using the Watson-Crick base pairing method to detect your RNA of interest. So basically you have to isolate your RNA from your cell choice or the cells that you have treated with some kind of drugs or anything. Then ultimately you run it in a gel, right? Just like any kind of blotting technique. After the gel is done, you cannot really use the gel to do a lot of hybridization steps, right? So you need a membrane. So you use specific membranes and you blot it, right? And this is your direction of movement. So from the gel, so from this particular gel, all of these RNA molecules get transferred into the membrane. Now you can do several stuff in the membrane. So this is how a RNA profile would look like. So basically you would have 28S rRNA, 18S rRNA. In between them you have a lot of mRNA, and then you have 5S rRNA. So this is how a typical RNA gel look like. Then what you're going to do, you put a membrane, you blot it, that we have already shown you. So in the nylon membrane you would use probe hybridization. So you would bake the nylon membrane, put it in the UV such that the proteins are nicely cross-linked to the membrane, and then what you are going to do, then you are going to add probe. Now all of these RNAs, sorry if I said protein, so all of these RNAs which are extracted from the cells are now nicely attached to these attached to this particular nylon membrane, right? Exactly. And there are specific particular membranes that are used for northern blot, which we don't really need to understand at this moment. But anyway, what we use to detect is radioactively labeled probes, or anyway labeled probes, not necessarily need to be radioactive, but you can use a biotin level probe or any other fluorescent level probe to detect it. So your job is to detect the RNA of interest. So you need to design the probe which has to be unique and picking up your RNA of interest. So then you flood your samples with your probes. Now some case the probe would bind, where it would find complementary sequences. So the probe might also bind non-specifically, for example in this particular case, to another RNA of interest. So that is why what you need to do, you need to get rid of all kind of non-specific interaction, and what you do is wash these membrane vigorously. As a result, non-specific interactions are gone and only specific interactions are remaining, right? And ultimately you know this particular probe was labeled, so you have a detection system. You can, if you are using a radioactive probe, so you can generate an autoradiogram to understand whether that particular mRNA is present or not. Or if you are using fluorescence based system, you can use another way to develop it, right? So this is all of the ways by which you can develop your northern blot. So northern blot is vigorously used to detect RNA of interest. Okay guys, so exactly this is how our northern blot gel look like. So this has quite a lot of usage, right? The whole goal is to understand whether a mRNA X is present in the sample or not. This mRNA X might be super interesting to you, or might be a disease causing gene that need to be expressed, or some kind of biomarker. So this is important. So you need to understand that using northern blot you are going to probe for your mRNA of interest, right? So this is the key goal for northern blot. So let me tell you frankly, people really use real-time quantitative PCR to answer the same question these days. They don't use western blot, they don't use northern blot technique so much, because what happens if in case of northern blot, if your probe is not so good, you could have non-specific interactions. Second of all, there could be other problems. For example, if you have very low amount of RNA, the probe might not detect it, or it might not be sufficient for visualization. So in that cases, people don't prefer northern blot as a technique these days. So real-time quantitative PCR is far more sensitive than northern blot, but northern blot also was developed in 1970s, and it revolutionized the way we look at mRNAs and gene expression profiles. So that is why northern blot is also important. But what is going to be asked in IIT-JAM, that again the name of the technique and the usage of the technique, possibly a scenario where northern blot can be used. In this scenario, we learned that northern blot can be used to detect your mRNA of interest, right? Or any kind of RNA of interest. So if you want to deal with RNA, northern blot is the choice. If you want to deal with protein, western blot is the choice. So these techniques are generally important for these kind of exams, because when you are going for MSc, you might need to use all of these techniques in your thesis project, or when you go to research, you go to PhD, you need to use these techniques. So having a sound understanding of these techniques would help you to choose or to frame your hypothesis or pursue your hypothesis. You can frame a hypothesis, but without the access to proper techniques, you might not design your experiments. So you need to understand bits and pieces of these techniques, how they can be used, where they can be used, and that could be really helpful for you guys to design experiment in future. So I hope in this video I am able to convince you about the about the usage, about the principles of western blot and northern blot. However, in this video we didn't get enough time to talk about southern blot. Maybe in other day we are going to talk about southern blot, and there are some other combination blot like some eastern blot, some southeastern blot, and all kind of these things. So we'll talk about a little bit of those things, not so important for IIT-JAM, but we'll just cover it up. But anyway, western blot and northern blot are super important, and we looked at their usage in biology, right guys? So I hope you understand this video. If you like this video, give it a quick thumbs up, and don't forget to like, share and subscribe Unacademy's YouTube live channel, because I keep on posting so many free contents in this channel. So if you don't have the capability of taking a plus subscription, you cannot afford plus subscription, hit that subscribe button. You're going to get free contents, guys, right? Again guys, I'm going to tell you that my plus courses are running for whoever is going to watch this video later on in the recording, or whoever has joined later. These plus courses would really help you to reach closer to your goals. These are highly structured courses, and by the way, you guys know that I was a IIT-JAM topper, right, twice in a row. So I pretty much have the experience that how we can study, what we need to study, and all of these things. So this structures course would really help you to take one step forward to your destiny. So I would really suggest you might think of joining the plus course. And if you're join for a long term, because if you join for a long term, the subscription is even lower, right? For example, it's like 800 rupees per month. Come on, 800 rupees, couple of movie tickets, right? Or maybe you spend more budget on fast food for a whole month than this particular 800 rupees per month. So it's super cheap, guys. Think about it. Anyway, if you have a specific comments, or if you have specific question, you can always ask in the comment section. Anyway, I provide nice visual handwritten notes and interactive flashcard which would really augment your learning, right guys? So don't hesitate to register today. So I at least hope some of you would join who are watching this video. If you like the explanation type, you like the visuals, you like the way we discuss together, if you think that these tutorials are very very interactive and not boring enough, then see you soon in the plus course. So with that, I'll stop this live stream.